All right, guys, this slide right here, we have seen in class lots of times. Oh, you can't see what I'm looking at. This slide is kind of summarizes evolution. You got to know that definition. It's a change in the allele frequency of a population over time. Individuals don't evolve, groups evolve. There's got to be variation, and we've talked over and over again in here about where variation comes from. It's the kind of thing they like to ask about on AP exams. What's the most important source of variation? Random mating, sexual reproduction. Random sperm, random egg. Mutations are a good source. Migration. What's another word or phrase for migration or immigration? Gene flow. All the meiosis stuff, crossing over, independent assortment, segregation. You got to have variation though. 2, there's got to be some natural selection. Some organisms have to be killed off before they reproduce. 3, the ones that live are the ones that are better adapted. They have to be genetic adaptations. They can't be what kind of adaptations? What was Lamarck about, the guy before Darwin?
Giraffes,
traits acquired.
Exactly, acquired traits. Traits that don't have anything to do with what? They don't have anything to do with what? With your genes, okay? You can't pass those on. Lamarck thought you could. To be important for evolution, it's got to be something related to your genes. And then finally, the most important thing is that differential reproduction thing. The best adapted have more babies. Their genes become more common. Their competitors less common. When we talk about fitness, I've said this 100 times in this class, it is not survival of the strongest. It's not survival of the biggest. It's survival of the ones that have what? Most babies. And I think there was a question on your multiple choice about that, right? Should have been an easy one, okay? Yeah, I mean, it's very doable. Population genetics is another topic we spend a lot of time on. Remember, again, populations evolve, individuals do not. When we're talking about populations, we're looking at the allele frequencies in a gene pool, all the genes of a population, gene pool. Remember, to be in the same species, there's a couple of things that are important. To be in the same species, you've got to be similar genetically, similar enough so that you can what? Make babies, and similar enough that your babies can make their own babies. They're fertile. A population are the same species in the same place at the same time. That's what makes things be a population. Now, Hardy and Weinberg stuff. You've got to be able to work Hardy-Weinberg problems. You saw in that test yesterday, there's probably 4 or 5 questions related to Hardy-Weinberg, right? At least 4 or 5. And there always will be. There'll always be 4 or 5 questions probably about Hardy-Weinberg. Those should be easy ones. And all those yesterday were pretty easy, right? Or wrong? Hardy and Weinberg were trying to describe the process of genetic equilibrium when evolution is not happening. Usually, they'll ask a question about these things. And there was 1 or 2 questions related to these 5 things, right? I think there was a question about drift, maybe. I don't remember what the question was exactly. But those are the 5 requirements for evolution not to occur. No mutations, specifically mutations in the germ cells. No gene flow. You've got to have a large population so you don't get chance changes in allele frequencies. Drift. Remember, there's 2 kinds of genetic drift. You need to make sure you know the 2 kinds of genetic drift. What are they? Yep, the founder effect and the bottleneck effect. The founder effect. Let's say that Sam here decided he was going to start a cult of Sam, and this cult was going to worship Sam. So Sam kidnaps 10 people. He takes them to a deserted island. And they have to worship Sam. And he will never let them off the island. They're going to live there for thousands of years, them and their babies. And it's the cult of Sam. Because Sam kidnapped these 10 people, kind of at random, he just grabbed the first 10 people he saw, Are they going to be representative of the human population as a whole? No. They probably got unique allele frequencies. That by itself is the founder effect, because their frequencies are different than the main population. But Sam also has some red hair, which is pretty rare, right? And let's say that Sam goes on to have lots of babies. And therefore, what's going to happen to the red hair alleles in that population? They're going to become pretty common. There's going to be a lot of red-headed people in the cult of Sam. Way more red-headed people in the cult of Sam than in the general population of the United States, or the world, for that matter. That's the founder effect. Yes? Red hair's a little complicated genetically. I think it is recessive, but it's typically dark and dominant over light. But then red is a whole different ballgame. We'll get into the genetics of that later. The other kind, the bottleneck effect, is when you have a big population and something wipes out most of it. So you start losing your diversity, and eventually you end up with just small, isolated pockets. And one of the big problems that causes is there's a lot of inbreeding going on. Whenever there's a lot of inbreeding going on because there's nobody else to breed with, you get a lot of genetic disorders, a lot of diseases. When you hear genetic drift, you should think small, isolated populations and frequencies changing because of what? Chance. Small, isolated populations and chance. Those are the things you should think about. Also remember, there's no natural selection and no sexual selection. Mating has to be random. We've got to make sure we can work a Hardy-Weinberg problem. So let's work this one just to make sure we can all work one. Remember, we got two equations we need to know. P plus Q equals 1, and P squared plus 2PQ plus Q squared equals 1. Remember that P is the frequency of the dominant allele in our gene pool. Q, the recessive one. P squared is how common are what kind of individuals? Homozygous dominant. 2PQ, the percent that are heterozygous. Q squared, the percent that are homorecessive. What do you always want to find first? Almost always, at least. Q squared. Because the thing is, if you know 20% are dominant, you don't know what percent of that 20% is hetero and what percent is homozygous, right? So if you find Q squared, those are the only people that show what phenotype? The recessive. Find that first. Find that first. So in this problem, they give it to us, right? The ability to roll the tongue is dominant over the lack of the ability. 49% cannot roll their tongues. That's the recessive phenotype. That 0.49, that 49%, is our percent homozygous recessive. So we got that. When it asks us to find the frequencies of the dominant and recessive alleles, what is it telling us to find? P and Q. You always need to find those. After you find Q squared, find P and Q. To find Q, we got to take the square root of Q squared. So Q is going to be 0.7. And that tells us in this population, 70% of the alleles related to tongue rolling are what kind of allele? Recessives. That's all that means. 70% are the recessives, which means the other 30% have to be dominants, right? So we just answered number 1.
Will it always list an easy number to find?
Yes. Now next year they're redoing this test so people can use their calculators. This year you cannot use calculator. So the numbers are going to have to be easy square roots. Or they'll leave it in terms of a square root like they did on that test yesterday. Yep. So number 2, give the percentage of the population that would represent each possible genotype. Find the percents. So the percent homozygous dominant, P squared. We got to square P. 0.3 squared is 0.09, right? 9%, 0.09. And 2PQ, 2 times P times Q, 42%, 0.42. So that's our answer to number 2. And don't just write the numbers down. Write out 9% homozygous dominant, 42% heterozygous, 49% homozygous recessive. tell people what those numbers are. And then the last one, if the population has 3,000 individuals, how many should be homozygous dominant? Well, these are our homozygous dominants. So we either need to go 3,000 times 0.09, or what I would do, I would go 9% of 1,000, which is how many? 9% of 1,000 is 90, and then just multiply that by, there's 3,000 people, so just multiply it by 3, and that'd be 270. If you have a hard time doing math in your head, just go 3,000 times 0.09 and work that out. Still going to give 270. Anybody have questions about how to work these problems? Got to be able to do Hardy Weinberg. Everybody can do it.
Did you grade that on last?
I'll try to get that back to you. Everything is graded now. A couple other things I want to review with you. Things you should have already seen. 3 patterns of what are called natural selection patterns. Some different terms for them occasionally. In stabilizing selection, nature is selecting which phenotype. The one in the middle. What's the fancier word for the one in the middle? Intermediate. If this is the way that humans initially were in terms of birth weight, quite a few small babies, quite a few middles, quite a few larges. Nature, though, decided, and I'm giving human qualities to nature. Nature wouldn't really decide. But nature decided to pick the middle because it was best adapted. It's what happened to these little bitty babies. They were sickly and weak, and they died of disease probably. These big babies literally couldn't get out. Nature was picking the middle. And over time, what happened to the birth weight of babies because nature was picking the middle? More babies started being middle weight. Now that's changing today because of what procedure? C-section. The C-section has shifted baby weight this way, bigger. Kind of the opposite pattern to stabilizing selection is disruptive. Now if you're using the outlines, they call this a different thing on the outlines. Anybody know what they call this on the outlines? Anybody got the outline here? That's okay. They call it diversifying selection. You don't want to know both terms. In disruptive or diversifying selection, nature's picking what? It's picking against the middle. Okay? It's picking the two sides. It's picking the extreme phenotypes. The example I used was of a butterfly in the middle. The normal butterfly was not poisonous. So what likes to eat non-poisonous butterflies? Birds. But the other 2 mutant versions of that butterfly, the ones on the outside, weren't poisonous either, but they were what? They were, the key word, they were mimicking, they were mimics of other poisonous butterflies. Birds aren't that smart, so they see an orange butterfly, and what do they think? Monarch, poisonous, don't touch it. And the other color, I think the other color is black. But regardless, it was mimicking another poisonous butterfly. So the birds didn't touch the 2 on the outside, but they like to eat the ones in the middle. So over time, the ones with the intermediate phenotype become less and less common, and eventually go away. And we end up with 2 separate groups. We said sometimes this disruptive selection leads to dimorphism. And that term, dimorphism, means 2 forms, 2 shapes, 2 kinds, something like that. And eventually what might happen to these 2 kinds, since now they're very different. Now they might become different species because they're sort of separating themselves out by habitat probably. And the last one of these patterns is directional. Nature's picking one phenotype over another in one direction. Here, nature's picking what? Big. Big is better. In the case of a horse at least, it's picking big. So over time, the horse and its ancestors became larger and larger and larger and larger and larger. Big is better, in this case. All right, key statement right here. To get speciation, gene flow must be cut off between populations. That's a very key statement. To get speciation, to get new species, you first have to cut off gene flow, which means you have to have at least two populations that are isolated, right? Separated. You're not doing what between those two populations? They're not mingling, they're not having babies with each other, right? They're reproductively isolated. Cut off. Before I get too deep into that, we talked about micro and macro evolution already. Micro are small changes within a population. Macro are mega changes, right? So big that they lead to new species. That's what we mean by speciation. So we might use the word macroevolution to mean the same thing as speciation. So what we're going to concentrate on for about the next 5 minutes or so are ways to cut off gene flow. One way, I know we've talked about it in this class before, is allopatric speciation. Yep, what did we say that word allopatric means? Different places. It actually means like different fatherlands or something like that. But today we use it to mean different places. Allo means different. So here we have two populations, same species, but they're separated by maybe a mountain, by an ocean, by a lot of distance, something that's keeping them apart. And because they're far apart, what's building up separately in those two different populations? Well, there is no gene flow, but what's happening in the two populations genetically? What always happens in populations? Well, yeah, but they have mutations, random mutations. Are the mutations in this population over here the same as the ones over here? No. So over a lot of time, how many mutations happen? A whole bunch. Most of those mutations are probably bad and just die out, right? But some of them are good. And are the good ones over here the same as the good ones over here? No. Because mutations are random. So given enough time and enough separation and enough mutations, this group becomes different from this group. And if you put them back together years later, what might be the case? Maybe we build a bridge over the river. And now they can go back and forth. What might be the case now? They may not recognize each other anymore. They may be a completely different species. They may be now so genetically different because of these mutations that they can't breed anymore. Okay, they're different. That's allopatric. Sympatric means what? Yeah, but what's it working? Sympatric. Same place. The example I used for sympatric speciation was frogs living in a pond. All look very similar. All living in the same pond, eating the same food, doing the same stuff. But the frogs mate all year long. They have breeding seasons, times. If some of the frogs mutated to go into mating season in March.
Due to the internet being down this morning, students will be coming to the classrooms to bring you a list of the students that will be excused to go to the blue gym at 8:45 this morning. Teachers are to keep the list. The list will be picked up on Monday. Thank you.
If you're already in them. All right, so what I was saying, if these frogs, some of them breed in March, and some breed in May. Even though they're living in the same pond, are they separated? Yeah, right? They're isolated. Therefore, there's no way. They can't breed. There's no way. There's no gene flow. And if there's no gene flow, what's going to happen given enough time? Mutation followed by speciation, new species. What I just described is temporal isolation, separation by breeding times. But there's some other ways this can happen. A lot of times in plants, and this is one I don't know that we talk very much about in here at all. In us, if we get an extra chromosome or a missing chromosome, what usually happens? Yeah, we die. Plants are much more tolerant of that. Plants can actually get whole new sets of chromosomes. Instead of being like diploid, they might become triploid. or quadruploid. And that's okay with plants. It doesn't bother them. They're okay. Sometimes what might extra genes mean for a plant? Sometimes if they have 3 sets of chromosomes instead of 2, what do you think that might make them do? Bigger, stronger, faster. Doesn't work that way in us.
Faster.
Yeah, grow faster is what I mean. Yeah, grow faster. So sometimes we say that plants can become polyploid. Maybe they cross with another kind of plant, or maybe just meiosis goes bad. And now instead of having diploid, now maybe they're triploid or quadruploid. Any kind of plant. But now, can a polyploid plant then breed with its diploid plant that it came from? No, because they're different. The chromosomes don't match up. So that creates a whole new species of plants. When they get extra sets of chromosomes, now they can only breed with other plants that have extra sets of chromosomes. So that's a new way to get a new species. It's separation by chromosome number. That's called polyploid if you have more than 2 sets. Kind of leave it there for Patrick. The other thing, and I'm not sure if we talked about this in here or not, these are different ways to isolate organisms. Cut off what? What do we kind of cut off gene flow? Some ways are called prezygotic. And there were some questions on your mock exam about this. And these are kind of straightforward. Prezygotic means they keep a zygote from ever being made, right? So, if they keep a zygote from ever being made, one way is habitat isolation. That's what we just described. Allopatric speciation, right? Different places. If they're in different places, they can't have any gene flow. Behavior isolation. Let's think about that for a minute. If a male bird wants to mate, he's kind of got to do a lot of stuff. One, he's usually got to be brightly colored, right? Because the female birds like bright colors. And if you're a male and you're not brightly colored, you may live a long time, but you're not going to what? You're not going to mate, because the females won't have anything to do with you. But that's not it. What else is a male bird in most species got to do to get a mate? He's got to know the right song. Okay? And if he doesn't know the right song, he's out of luck. Okay? What else? He's usually got to do either a dance or some kind of aerial fly pattern, crazy stuff, like zipping and twisting and turning. And if he knows the right pattern, that impresses the females. If not, he's out of luck. That's what we're talking about here. Behavioral isolation. If you don't know the right moves, if you don't have good moves as a male bird, no ladies. Okay? That's what we're talking about. You got to have the right moves. Temporal isolation we already talked about. Time. Separation by mating season. Mating time. It's important. 4 just kind of makes sense. Mechanical isolation. Maybe they mutated so the parts just don't fit anymore. Parts don't work. The reproductive parts don't go together. And then the last one, gametic. Maybe the sperm and egg do make it to each other. But once they find each other, they don't really know what to do with each other. They can't make a baby. They just don't work anymore. So these are things that separate 2 species from being able to make a zygote at all. If you can't make a zygote, you are what? If you're 2 different animals, you can't make a zygote, you are reproductively isolated. Hello? Hey. I don't have her today, or I would. We're on a B schedule today. Yeah. We had, since we had all those interruptions yesterday, we're doing like a Tuesday, Thursday day today. Sorry. All right, bye. So these are ways to reproductively isolate 2 groups of organisms so that they can't even make a zygote. They may still try to mate, but they're never going to make a baby because it's not going to work. The next way, the next group, oops. Can I put what? Oh yeah, Elmo's always in the way. The next group are post-zygotic, which means post-zygotic means, yeah, they get together, they have sex, they make a baby, they make a zygote. But what happens with that zygote? It either dies or it's sterile. Okay, one of the two. That's so. Even though they make the baby, they're still pretty much what? Different way. Different species, because it's like this. The post-zygotic things would be like a horse and a donkey. Horses and donkeys are really closely related, right? They look a lot alike. They act a lot alike. So much alike genetically that they can make babies. And their babies are strong and big, right? Mules, that's why we make them. They're big, strong animals. But what's the problem with donkeys? They're sterile. So even though horses and donkeys can make babies, they're still different species because their babies can't make babies. Oh, donkeys can mate with donkeys. We're talking about mules. A horse and a donkey's baby is a mule. A mule can't have babies. Mules are sterile. I think they are, but I'm not. Don't quote me on that. I'm not sure. What now? Ligers are real. Yes.
It's actually called a different thing. Like the male and female is called a tygon.
Right. It depends on who's the mom and who's the dad.
Tiger's my favorite animal. I want to see.
All right. The post-zygotic barriers. We just mentioned what they were. Two different groups can get together and make a baby. The zygote is made. The zygote is made. But one of the post-zygotic barriers is called reduced hybrid viability. That word viability is a word we need to think about. Ability is part of that word. What's vi? V-I or V-I-V. We've seen that several times in this class. Viv, what does that mean? Live. Ability to live. That's what viability means, right? Ability to live. Where else do we use viv at? Viviparous. Animals like us that do what? Live birth. No eggs involved, no hard-shelled eggs at least. Live birth. In French, what's this phrase "c'est la vie" mean?
C'est la vie, like.
Well, sometimes you use it that way. That's not really what it means, though.
It means like live life.
It means that's life. That's life. C'est la vie. And there's something similar in what's the Spanish word for life?
Viva.
Viva, right? Viva, like Viva Las Vegas, right? Regardless, reduced hybrid viability. They can make a baby, but it dies. They can make a baby, but it dies. Reduced hybrid fertility. Yeah, these two different groups get together, they make babies, but their babies can't make babies. So is that ever going to lead to anything fruitful? Probably not. And then the last one, maybe for a generation or two, the hybrids can have babies. But then over time, their genes, something goes on with their genes and they become sterile. Call that hybrid breakdown. I think that's more common in plants. That for a generation or two, everything seems to be working, and then things kind of go bad genetically.
So like, if there's some species that the hybrids are born like really, really weak, would that be?
That'd probably be this one right here. So we're talking about ways to separate two groups of animals. How are lions and tigers separated? I don't know if a liger is. I don't know if let's just say a liger was fertile, and I don't know if it is or not. But if a liger was fertile, lions and tigers are still separated. How are they separated? They live in way different places, right? Lions and tigers don't live close to each other. They're habitat isolated. They live in different places. It's like allopatric speciation. They're in very different parts of the world. They're never going to run into each other in the wild, lions and tigers. Okay. All right. This slide, I'm not sure that we've looked at it in this class at all or not yet. So let's talk a little bit about this. We know what speciation means, the creation of new species. There's two kinds of speciation that you need to be familiar with. And I know they talked about this some in the Saturday session, and I think maybe we might have talked about it a little bit in class. As soon as we see that word Genesis, what should we be thinking? Nope. Creation. Now, what does "and" mean, though? No creation. When in anagenesis doesn't really make a new species. In anagenesis, you have species, and yeah, they may mutate and they may change over time, but it's still at the end what? One species you started with, and still at the end you still got what? One species. They might have undergone some microevolution, and they may look a little bit different at the end, but it's still just one species, right? So this says the accumulation of new changes over time within a species leads to a new species. But you started with one, you still got what at the end? One. Cladogenesis is different. What do we know a clade is? A group. So it's the creation of new groups, right? New classification groups. So here you start with one species, and what happens to your phylogenetic tree? It branches. This is a phylogenetic tree branching out, creating new species from one that you started with. So here it says branching off of an existing species occurs because of moving into new environments, reproductive isolation, or other environmental stresses. What you need to know here, creation of new groups. One species evolves into multiple species. That's the kind of thing you need to know. Creation of new groups. And again, this PowerPoint is posted, and the outlines are posted for all this evolution stuff. So use those things. Put them up there for you to use. The creation of new classification groups. Cladogenesis.
Is that the second type of speciation?
Yep, anagenesis, cladogenesis.
Are we going to do a cladoworm in here?
What now?
Are we going to do a cladoworm?
Yeah. We'll do one eventually. All right, next here, this is showing you anagenesis. How that you started off with this bird, and over time, over a gradual accumulation of mutations, this species of bird changed into this one. But you started with one, you ended up with one. That's anagenesis. Here in cladogenesis, you started with one species, but your family tree split into new groups, and you ended up with more than one.
Wait, can you repeat what cladogenesis is?
Start with one species. That species evolves into multiple different species, different groups. One species, the phylogenetic tree branches into several different species. Creation of new groups, cladogenesis. All right, not going to say much about this slide. Biogeography is a term I know they talked about Saturday. It's kind of a simple idea. Biogeography is where on Earth are things found. Literally, what does that word mean? Biogeography.
Study of life.
Places where there's life.
Where is life found on Earth? Right? Geo means Earth. Okay? Where is certain living things found on Earth? That's biogeography. The reason we want to mention this is this idea of convergent evolution. And instead of looking at this slide, I want us to look at this picture. You got these two animals. They're flying squirrel-like things. This flying squirrel lives in the western part of the United States. This one lives in Australia. This is a placental mammal. I've said that wrong. This is a marsupial. This is a placental. Are they very closely related at all? No. But they look what? They look almost the same. They have very similar characteristics. Usually, when they look the same, that implies common what? Common ancestors, common DNA, right? But here, that's not the case. Why do they look the same here? Partly chance. but mostly because where are they living?
Same environment,
The same basic environment. Even though it's on two different continents, where these squirrels live and where these live, it's almost the same kind of place. And to succeed in that kind of place, what do you need? That kind of shape. You need to look that way. You need to be that way. That's called convergent evolution. What's the word converge mean? Converge. Come together, right? So it's when two unrelated organisms evolve to do what? Two unrelated organisms evolve to look very similar because they live where? In similar environments. Are they what?
Where are flying squirrels at?
I'm not sure. I've seen them, I don't know if any live around here or not. I don't even know. No idea. I don't know much about flying squirrels.
We should get.
Are those little squirrels that might come in the ball?
Those are probably the sugar gliders, yeah.
How small are they?
How come we can't have flying squirrels?
I don't know. I mean, we could. They probably could live here. They're just not. This is not where they happen to evolve.
Kind of hard to travel.
Yeah. They're cool. They're cool. Back to what I was saying, though, guys. I'll get you in a second. Another example of convergent evolution: shark and whale. Shark and whale. No. Shark and whale. Okay. Are sharks and whales closely related? No. You got a fish and a mammal. Very different genetically. Not much related at all. But if you were swimming out in the ocean and 50 yards away you saw something big, you probably wouldn't know if it was a shark or if it was a whale, or a dolphin, because they look so much alike. Okay, and the reason they look alike is not because of homology, not because they're genetically similar, but because they live where? In the same place. And what does that environment dictate? That they have that shape, right? To be able to make it through the water, you've got to be shaped like a bullet. You've got to have fins in certain places so you can steer and accelerate. That's convergent evolution. Two organisms that are not closely related look a lot alike because they live where? The same kind of environment. And the environment dictates that they look that way. Converge means to come together. Two unrelated things become alike because they're living in the same kind of place. Is that what you were going to ask me? Drew, were you going to ask me?
Like the predatory whales, like killer whale and the whale shark, like do you know if they're actually evolved from whales or are these kind of their own thing?
I think the idea is that they all evolved from whales. That there's all a common ancestor. Now the whale shark's not predatory though. Whale shark is it's a filter feeder.
Oh.
It's just really big. It's just a filter feeder. It's actually, even though it's a shark, it's a fish. In terms of the way it eats, it's more like a whale. Manatees are just mammals.
Do they have any common ancestors? Mermaids?
Some kind of mammal. Yeah, it's hard to say. I don't know. I don't know enough about it. What about they're definitely related. How closely? I don't know. All right, back on this, guys. Back up here. Couple of words up here you need to make sure you know the definition of. Converge, we said two unrelated things become alike due to similar habitats. That's converge, coming together. What's diverge mean? Go away from each other, right? So, yeah, they're related. But in divergent evolution, you have a group of organisms, and that's very closely related. But you put them in different places, what happens? You have a group of the same population, same species. You take them and you split it in half, and you put one group over here, maybe in a cold place, and you put one group over here in a warm place. What happens in just a few years?
The cold ones grow further.
Well, they evolve to be very different. That's divergent. So diverge, you have closely related organisms in different environments. Therefore, they evolve to become what? Very different. Diverge, become different. Converge, become the same.
Tell us about deers.
Probably so. Yep. An important term that I know you've heard on the Saturday sessions, and I know you've heard from me. A very important term to note: adaptive radiation. That's a term that AP people like to ask about. I don't think there was a question about it on this AP exam that you guys took yesterday, but there usually is something about adaptive radiation. Adaptive radiation is when you take a group of organisms and you put them in a new place, a new environment. When you put those organisms in that new environment, what happens? A good example of this is Darwin's finches, the birds. You put these Darwin's finches out on the Galapagos Islands. They're probably the first birds out there. They're very different. There's not a whole lot of food out there. So when you put this group of birds out there, most of which like to eat the same food to begin with, what did those birds do very quickly? They evolved. They evolved very fast. They speciated very fast. That's adaptive radiation. Very fast speciation. So that the organisms speciating they're speciating to avoid what? Okay, but what? Give me another word. Things evolve in biology oftentimes to avoid competing with each other, to avoid competition. So let me give you a good definition of adaptive radiation. That one up there is a good one, but I'm going to give you one a little bit different than the one up there. We're going to say in adaptive radiation, you take a population of organisms, you put them in a new environment. Once they're in that new environment, they very quickly speciate in order to avoid what? Competition, and in order to fill what? Fill new niches that are available to them. That word niche, important word. Niche. I know we've used that word in here before. A niche is a role where? In the environment. It's kind of what you eat, where you live, what you do. It's your place in nature, your niche. So things evolve to avoid competition and to fill new niches. Adaptive radiation. Fast evolution when a population is put in a new place. Fast speciation. So that these things avoid competition and fill new niches that have been opened to them. Now I'm going to tie that in with something else that I know they talked about in the Saturday thing, and we talked about a little bit in here. And the terms I'm going to tie that into are gradualism. And what's sort of the opposite idea about evolution from gradualism? You guys don't know. I know there was at least one question on your mock exam about this. Punctuated equilibrium. The idea about gradualism, this was Darwin's idea about evolution. That evolution happens slowly by the gradual buildup of what? What makes us change? A buildup of what? But what gives us those alleles that are different? Where did all new alleles come from? Mutations. Okay? Darwin's idea, and the people right after him, was that evolution happens slowly because of a gradual buildup of mutations. Is that the way evolution happens? Sometimes, but not all the time. This idea of punctuated equilibrium is based on what evidence? Maybe you don't know. A guy named Jay Gould came up with a theory of punctuated equilibrium. Exactly. Jay Gould is still alive. I think he works at Harvard. He was looking at fossils. Fossils are made mostly in what kind of rock? Sedimentary. So the deeper you go, what does that mean about the rock? Older, because it's layer on layer on layer, right? Sedimentary means it's in layers. So Gould went down, started looking at rock layers, and he found for periods of time, layer after layer after layer after layer after layer had basically the same kind of species in it. Very little change. Layer after layer after layer, constant. That's equilibrium, right? Equilibrium means not much change is going on. For layer after layer, almost no change. And then he hit a layer in the fossil record where what happened?
It was punctuated.
Huge change, a punctuation mark happened, okay? Where there was mass what? Mass extinctions followed by mass what? Mass extinctions followed by mass new species appearing, right? And what did he think caused that to happen? What caused his punctuation marks? Some kind of bad environmental thing going on, like a meteor hitting Earth, okay? An ice age, a huge outbreak of some kind of disease, some kind of environmental disaster. When that environmental disaster hits, most things die, right? Before the mass extinctions happen. And when all those things have died, now what's available to the things that live? New what? New niches that nobody's filling. And what do we say happens when there's new niches? New species evolve to fill those niches, to use those resources. That's evolution. That's what we're calling here punctuated equilibrium. Think about it. It makes sense. If you're an organism and you evolved to be very good in your environment, and your environment is staying pretty much the same, does it make sense for you to do a whole lot of evolving? If you're good at living in that environment, and that environment is staying the same, does it make any sense for you to start evolving? No. Okay. So that's what we're saying. That's the equilibrium part. If you're good at living in your environment, ladies, take this please. It makes no sense to change. So there's long periods of stability in the fossil record, followed by these punctuations, these fast, huge changes because of some kind of environmental disaster. Comet hitting the Earth, disease outbreak, ice age. So this punctuation was a mass extinction event followed by mass speciation. And maybe the best example is the whole dinosaur bit, right? We think a meteor hit the Earth, killed off all the dinosaurs. Some of the things that lived were mammals. And these mammals before, they think, mammals were around the same time as dinosaurs, right? Some mammals, okay? Some mammals. But what do they think the mammals were like when the dinosaurs were around?
Very small, bro.
Small, like, insect, like a mouse kind of like animals. Okay? And then over time, insect eating is what we're saying. But when the dinosaurs died, what was now available? Lots of new niches. And the mammals evolved to take in those niches. The dinosaurs had been the big dominant predators. They weren't there anymore. So now what evolved to become the big dominant predators? The mammals. Okay? That's what we're calling punctuated equilibrium. Definition. Let me put a slide up with a good definition of it. Theory in which long periods of little evolutionary change are followed by brief periods of great changes. That's punctuated equilibrium. And no, go ahead. Go ahead.
So with fast evolution, with punctuated equilibrium, would that go under macroevolution since it's such a big change?
Any kind of speciation goes under macroevolution. Anything that leads to new species. So gradualism, theory that evolution occurs slowly through the gradual accumulation of mutations, and punctuated equilibrium, the theory in which long periods of little evolutionary change are followed by brief periods of great change, usually because of some kind of huge disaster on Earth. Mass extinctions, that opens up all kinds of new niches, followed by mass speciation. Let's think back to human history. So kind of a related kind of thing, the Renaissance. Back in the Renaissance. You guys know what the Renaissance was, right?
No.
What sort of sparked the Renaissance to happen?
Like the church reformation.
Nope, before that.
Which one?
The Renaissance, the great Renaissance with Da Vinci and Michelangelo and all those people. What sparked it to happen?
All
The
the
Black
signs.
Death. Okay? The Black Death. Because here was the thing. Before the Black Death, most people were poor. There wasn't a whole lot of resources to go around. The Black Death comes along and it kills a huge percent of the population. Now there's a whole lot more way. There's a whole lot more niches. There's a whole lot more money. There's a whole lot more resources because their stuff's still there. And the people that live just go take their stuff, right? It's the same kind of thing as we're talking about here. Some big environmental change comes along and that changes the world. It causes mass changes. The plague caused the death of many people, but the people that lived were now way more rich than they were before those other people died. Because now they took their stuff. No. The Black Death is still around.
What?
Yeah. Black Death is still around. It's just not, it's not that huge of a deal because it's a bacterial disease, which means we can treat it with what? Antibiotics. The plague. Yeah, the Black Plague. Okay? All right, moving on a little bit here. Let's see if we can't finish this stuff up. Remember, these things are called phylogenetic trees. Join me. Phylogenetic trees or cladograms. And that the fewer branches there are between two groups of organisms, what does that mean? The more related they are. At every Y in this branch, what's implied to be there? Like a common ancestor. So right here is a common ancestor between these little dinosaurs and birds. This phylogenetic tree is trying to show you that birds are on the same family tree as dinosaurs.
I think that's body on two legs. Don't
I mean, same way we do.
Yeah, but we're like straight up. They're like,
Well, that's what the tail is all about. It's like a seesaw. So the tail's a counterbalance. If they didn't have a tail, they'd have a hard time balancing themselves. I mean, think about it. Think about it. It's a lot like a pregnant woman. When you ladies get pregnant, your balance is going to change. Okay? You watch these ladies; they get this big weight in the front, and to be able to walk, they walk like this, right? Okay? That's the counterbalance. If they tried to walk like this, they would fall. Okay? They have to walk like this. Okay? I mean, even these guys with the big beer bellies, they walk like this, right? It's like they're sticking it out, showing it to you. They're proud of it. That's not what they're doing at all. They're walking that way because otherwise they would fall on their face. Okay? It's counterbalance. Yes. Sure. One quick thing, guys. Back to this cladogram stuff. And everybody who was here Saturday, you had to draw a cladogram.
Yeah.
What do you put in the bottom left of your cladogram?
The lowest.
The most primitive organism, right? The one that has the least of the traits that you happen to be looking at. Okay? What do you put in the top right of your cladogram? The most of all, the one with the most derived characteristics. The ones with the most evolved characteristics. And then you go back down to your bottom left, and you find the one, not the one with the least of the traits, but the one with the next least traits, right? Draw off a branch. You go from there. I think you guys can probably handle that. Here's a phylogenetic tree for vertebrates. You need to know the order. Actually, this is just reptiles. I want to look at the one for vertebrates. Here's the one for vertebrates. Phylum Chordata, vertebrates, right? Phylum Chordata. Remember, there are two groups of chordates that are not vertebrates. These two guys over here, the lancets and the tunicates. They have a notochord and gill slits and dorsal nerve cord and all those kinds of things, but they don't have that. They don't have a backbone. They don't have a vertebral column. According to lots of different pieces of evidence, the first vertebrates to evolve were fish. And then over time, the fish gave rise to the amphibians. The amphibians gave rise to the reptiles. And the reptiles gave rise to birds and mammals. Okay? And we see the big jumps, the big adaptations that allowed them to do that. We've seen this same slide when we talked about vertebrates before, right? Couple of key important points to make sure you know about. One of the themes Saturday was this idea of homologous features. That should have showed up in every session Saturday. Homologous features. You know homologous means the same, or at least similar. A good example of homologous features are like the arm of a person, the flipper of a whale, the wing of a bird. If you peel off the skin and the muscle and everything on top of the bones, and you look at the bones in all three of those structures, they look basically the same. And why do they look so similar? We're all related. We all have common what? Common ancestors and therefore common DNA. The ultimate homologous feature is the same way. Having similar way. DNA. That's the ultimate homologous feature because that's responsible for all the others. Things have homologous features because they have similar DNA. They're related. They have common ancestors. So if two organisms have a homologous feature, that should tell you they're related. They're evolutionarily related. Now, an analogous structure, people confuse those two, homologous and analogous. Homologous means they're the same because of similar what? Yep, and similar DNA. When you hear homologous, think similar because of DNA. Analogous means that two things have the same function, but not the same DNA. Like the wing of a bird and the wing of a mosquito. They both make things fly, but are they made at all the same way? No. Birds have feathers and bones. Mosquitoes, titan. No feathers, no bones, made a very different way. Just because they both have wings, does that mean that birds and mosquitoes are very closely related? No, they're not. It's two different solutions to the same way. The same problem, flight, okay? Two different ways were evolved to fly. The bird way and the insect way. And they're both very different. Birds and insects are not closely related because they both can fly. That's not a homology, that's an analogy. When you give an analogy, what is that? Just in English, what's an analogy? It's when you compare two things, right? So yeah, they both can fly, but can they both fly because they're related to each other? No, it's two different solutions to the same problem. So realize, if things have homologous features, they have them because they're related. Same DNA. If they have analogous features, not related probably. Two different solutions to the same problem. Also make sure you know what vestigial organs are. Vestigial structures are structures in an organism that seem to have no function, but did in what? In ancestors, way back. Okay, like our appendix, like what else in us?
Tailbone.
Tailbone, and like what in whales?
Pelvis.
Pelvis and little leg bones. They have little leg bones. They don't work. Wisdom teeth, maybe. Yeah. So,
Spleen wouldn't be considered, so even though we can live without it.
what now?
Spleen wouldn't be considered.
No, spleen has a function. It does have a function. You can live without it, but it does have a function.
Is it gallbladder?
Gallbladder has a function too. You can live without it, but it does have a function.
What's your gallbladder?
It stores bile from your liver.
What's your spleen do?
It filters blood.
So your appendix does literally nothing.
As far as we know, it doesn't do anything. Just sitting there.
Oh.
All right. A couple of other things that people use to back up the idea of evolution is something called comparative embryology. And that's pretty self-explanatory, right? Comparing embryos. Comparing embryos, looking for similarities and differences. I'll come back to this slide in a minute. Here's the homologous features we were talking about before. All very similar bone structure between all these limbs, appendages. Yep. Now here's a slide of comparative embryology. We're looking at the embryos of a lizard, a tortoise, a pig, and a human. And notice, we all start out, unless you were an expert on embryology, during the first few weeks of development, you wouldn't know the difference. It'd be hard to tell. We all have a tail. That's a homologous structure. We all have gill slits. That's another homologous structure. We go down to the next stage of development. Notice they're still all pretty similar. It's hard to tell them apart. Now, when we get down to the third stage, kind of different, but this should show you in this third stage that we're more closely related to a pig than we are to these other things, right? The tortoise and the lizard look a whole lot alike. The pig and the human look a whole lot alike. Here's the thing. Join me. Comparative embryology. Looking at the way embryos develop. The more similar the embryos are, scientists would say, the more what?
Related.
The more related, the more similar the DNA is between the two things. The more related they are through evolution.
So turtles are more efficient.
Turtles are what? Yes. Yes.
They're just shells.
All right, the other big idea I want to touch on is this. Go ahead.
Once they're shells gone.
Oh, yeah.
They're gone.
How come it makes so hard?
But it's got calcium carbonate. It's like bone, similar to bone. Artificial selection is something else I want to make sure I touch on. And this is something you should have talked about Saturday as well. Almost all the crops that we eat today were artificially made. Pretty much all the breeds of dogs that we have today are man-made. Dogs are a man-made thing. Okay? Dogs were not originally on this earth. Men created dogs. And we created them by selectively breeding different kinds of wolves or foxes or something like that, right? They came from wolves or foxes or coyotes or some similar kind of wild dog-like animal. All these breeds of dogs we made by taking two dogs that were similar to what we wanted and breeding them. And then take the babies that were more like what we wanted and breed them together and breed them and breed them and breed them. That's why these dogs that are purebreds have a lot of what? Genetic disorders. Because they were made by us by forcing what? Inbreeding. Okay? Artificial selection. These big old tomatoes that you can go buy in the grocery store.
They've been breeding with their
Okay. Are these big old tomatoes you buy in the grocery store, are those natural?
No.
No. We made them. Okay? We found the wild tomato with the biggest tomatoes on it that we could find. And we did a lot of inbreeding. And we just kept breeding. We kept selecting for what? We kept selecting for large. Breeding, until we got a plant that consistently made a really large fruit.
That's weird. The bulldogs is nothing.
No, not really. I mean, we've been doing it for generations without really even knowing what we were doing. Wheat. Wheat's not real. And I know I said weird, that's okay. Where did wheat come from? Wheat's not natural. We bred it. We bred different grasses together to make those big grains that we get with wheat. Okay? That's artificial selection. That's important to know.
If you grow wild, is that just from?
Well, grasses, but the original wheat we created by breeding different wild grasses together. Yes.
It said that dogs, they were mixed in with cats.
I don't know about that. I have a hard time buying that. Don't know for sure about that. All right, last couple of things and we'll be done with evolution. Phylogeny, phylogenetics. We talked about that word all year long. It's the study of evolution, right? Phylogenetics. Here they say the evolutionary history of a species. That word you got to know. As soon as you hear phylogenetics, think evolution, 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 evolution. Phylogenetics. If you hear us talk about molecular phylogeny, we're looking at the evolution of molecules, right? Evolution of molecules. Molecular phylogeny. I want to show you an example of that. You don't need to write all this down. Just hear the word phylogeny. No, it means evolutionary history of a species. Molecular phylogeny, you're looking at the evolution of molecules. And I want to show you a picture of what I'm talking about here. In this slide, we're looking at the structure of hemoglobin. Hemoglobin. And we're looking at the amino acids in one of the polypeptide chains of hemoglobin. How many polypeptide chains does hemoglobin have? It has 4. Okay. We're only looking at one of them. We're looking at the order of the amino acids. In this particular polypeptide chain, there's 146 amino acids. When we compare a human and a monkey, there are only 8 differences out of 146. What does that mean about humans and monkeys? Our DNA is very, 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 very similar. Okay? Almost exactly the same. When we compare humans to mice, there's 27 differences, but that's out of 146. So that means with humans and mice, we're still really, really close, right? Our DNA is very much the same, at least for hemoglobin. Humans and chickens, only 45 differences out of 146, so still pretty close. Okay? Humans and frogs, a little bit different, 67 out of 146. Humans and lampreys, way different, right? We're not very closely related to lampreys at all. This is molecular phylogeny. Molecular phylogeny. Looking at molecules, hear me on this, looking at molecules across species and comparing them. The more alike those molecules are, what does that mean about the organisms that have them? The more related they are. Okay, so we're more related to monkeys than we are to lampreys. We're more related to chickens than we are to lampreys. Lampreys are more related to frogs than they are to us, right? That's molecular phylogeny. That's one example. Hemoglobin. Another example is this right here. This is cytochrome C. And you had a question, very first question on that mock exam was about this, right? Cytochrome C. Cytochromes are proteins found where? Where are cytochromes found? The electron transport chain of the mitochondria. Okay? And in photosynthesis. We're talking about animals right now. So, important in electron transport. You didn't need to know that to answer this question, though. All you needed to know to answer this question is this pattern. Here they're looking at the amino acid sequences inside this protein. Between humans and monkeys, notice there's almost no differences. Between humans and pigs, there are very few differences.
Where did you say cytochrome C was again?
It's in the electron transport chain.
Okay.
Humans and turtles, there aren't very many differences. So the more alike two molecules are across species, the more related those species are. This pattern is the same pattern we would find in the fossil record of vertebrates. What vertebrates supposedly came first? Fish. What vertebrate supposedly came second? Amphibians. Notice we don't have an amphibian up here, but if we did, the amphibian would be right in here. And then what came next, supposedly? Reptiles, and then what? Birds and mammals. Okay? So, there's all kinds of things that back up that idea of vertebrate evolution from fish to mammal. That's molecular phylogeny. Looking at the molecules in living things, comparing them across species. The more similarities there are in these biomolecules, the more related the organisms probably are.
Is it a lamprey like?
It's a jawless fish.
Okay.
Jawless fish. Here, this is looking at a bird wing and an insect wing. Very different. Does not imply what?
No.
Does not imply common ancestors or common DNA or relatedness, because they're different. Talked about artificial selection. Don't go deal with that today. We did that. Talked about this. Not gonna worry about that. I think we pretty much covered what we need to cover with evolution. So that being the case, here's what I want you to do. I want you to get this handout in front of you. This one right here.
Let me see.
The one that's two-sided. Yes, that's it. Get the two-sided one in front of you.